So welcome to another podcast from Odell Technology. I'd like to introduce you to Soren Breinholt from Alligator Biosciences. Hello, Soren.
Hi, Stephen.
It's wonderful that you could join us today, and thank you very much. Soren, I think you have one of the most exciting bioscience companies in Europe right now, and I was wondering if you wouldn't mind telling us something about yourself and how you ended up where you are today, please.
Yeah, happy to, Stephen. So, as you said, my name is Søren. I'm a Danish citizen. I'm a Danish citizen, grown and raised in around Copenhagen, and did my education there at University of Copenhagen. I did my PhD there. That was great times. And then I had the fortune to be able to do my postdoc in Paris for a couple of years at Pasteur and also Hôpital Necker. I learned a lot from those international environments there. Got back to Denmark to join Novo Nordisk, at that time a medium-sized pharma. Since it has developed into being one of the leading pharmas globally, and I had there over two stints the opportunity and fortune to work with very, very intelligent and clever and forceful people that have taught me a lot about how do you carry yourself in this environment, how do you develop drugs, and how do you run a company. And in between that, I had the opportunity for almost a decade to work in another biotech company called Symphogen, where I also worked with highly intelligent and engaging people. And I think those 25 years in combination has given me a unique view of the industry, what really drives it, how the focus on patients is the ultimate driver, but also how all the components of the business and the environment, the microenvironment around biotech and pharma sort of works together. So I've been so lucky to have been touched by so many gorgeous people during my career.
Oh, that's a wonderful thing to say about people. I like people who praise others and realize we're all a team. So, Soren, tell me a little bit about, or actually, could you tell me a lot about Alligator Biosciences, please?
Sure. Yeah, so Alligator is a global biotech company located in Sweden, in the southern part of Sweden, in the city of Lund. The company was founded in 2001, so we have a couple of decades of experience under our belt. We are uniquely focusing on developed so-called antibody therapeutics, antibodies that stimulate the immune system, so sort of enhances the body's own ability to fight, in this case, cancers. And we do that with a small team here of around 50 people, covering everything from the very early discovery to clinical trials. And then we sort of gear that capacity and capabilities by working with different contractors on a global scale. Yeah.
I think that I heard about you firstly through pancreatic cancer and metastatic pancreatic cancer. Can you tell us something about that?
Yeah, I can. So that is one of the most horrific oncology indications. It's an indication that is unfortunately diagnosed very late in these patients. So when people get this diagnosis, many of them have already metastases. So that's sort of the tumor spreading to other organs than the pancreas, and many of the patients have liver metastases, and that's really not good. So the prognosis for these patients is really bad. On average, in oncology, we talk about 5-year survival rate. So how many of the patients are alive 5 years after a diagnosis? And in metastatic pancreatic cancer, that is less than 5%. And so that is in itself is really some hard numbers. It's approximately 200,000 cases in the US and Europe every year. Approximately 400,000 globally. It is estimated to become the second deadliest, or sort of the cancer that leads to second most fatalities on an annual basis before the end of this decade. And you can see for the patients when they get this diagnosis, the treatment options are actually pretty few. So only about 20% of the patients are offered surgery, either because they have metastases in the liver or other places, or because the tumor is located in the pancreas, so it's inoperable. So for the last 80% of these patients, so that's more than 150,000 patients in Europe on an annual basis, that's basically only chemotherapy as a therapeutic options. And there are different types and different regimes of chemotherapy, so cell poisons basically that are offered to these patients. But even today, it's approximately only 1/3 to 1/4 to 1/3 of the patients, maybe in some cases up to 40% of the patients that actually have a clinical benefit from these chemotherapies. And when we talk clinical benefit, what we really as a medical community can offer these patients is to maybe increase their life expectancies from 3 to 4 months to maybe up to on a median range 11 months. So it's a disease we've known for a long time. We've not really seen any medical breakthroughs over the last 20 to 30 years, and there is a significant unmet medical need. Thursday, November 16, was World Pancreatic Cancer Day, and I can only sort of encourage everybody to get in touch with your local patient organization and see if there's anything you could do to help these brave people help us fight this disease.
That's a wonderful thing. I wasn't aware of that. Thank you. With regards to the tumor-directed immunotherapies that you're working on, it always fascinates me how an organization like yourself can work with the health technology assessment organizations in Europe and come up with meaningful reimbursement and how drugs are paid for of this caliber. Can you tell me something about that? Because I think that's an enormous body of work that you undertake.
But I mean, first of all, it's about developing your drugs so that you can ensure that it gets to the patients as quickly as possible. And basically, and you know this very well, Stephen, that you need to find drugs that are at the same time efficacious, so it gives eventually added quality life to the patients. But at the same time, do not give so many additional side effects that these additional months or years to live are sort of not worthwhile quality-wise.
Absolutely.
And when you sort of secured that you've done that, and then in your phase 3, it's really about ensuring that the data that you have shows you exactly that. And then starts the works with the health technology assessment organization across Europe on really ensuring that you can, I'm sorry to say, put a price on that additional quality life that you offer the patients here. And you can say that takes a lot of early discussion. It takes a lot of data. And of course, different diseases are different. In pancreatic cancer, there is nothing other than chemotherapy. So you can say on everything other thing being equal, the bar is somewhat lower. In other indications, there are alternatives. Here, for us, it's basically a manner of ensuring that we have as many patients responding as long as possible, because in pancreatic cancer, you don't really have that biomarker of disease progression or diagnostic biomarker that allows us to, at least at this point in time, to stratify patients in the clinic to say these patients are eligible for the treatment, and these patients are not eligible based on a very sound molecular criteria. That is, of course, something that we are striving to find and develop, but that is tough in these diseases that have several sort of genetic underlying reasons for developing.
I've seen the advent of some epigenomic testing in America of new diagnostics for various cancers, and I'm not aware if there are yet the indications for use for pancreatic cancer, and that could be a game changer.
Unfortunately not. It's clear that the whole field is looking at some of the very clear biomarkers. For instance, in pancreatic cancer, many of the patients have mutations in the so-called Kras gene, and now, as we are getting insight to some of the recent big clinical trials, we can see that for certain of the chemotherapies, there might be, what can we say, preferential responses in some of the mutations to some of these chemotherapies. And our hope is, of course, also with mitazalimab, our lead candidate, that we can delineate a clear genetic profile of responders and non-responders. But unfortunately, we are still, I think, some years away from that.
So I hope that's the situation. With regards to vaccination for cancers that's going on, the National Health Service in the UK recently signed a significant deal with a German organization. And I know that other governments are looking at new vaccines for cancer. Yeah. Is that something that you're involved in?
We are not directly involved in that. You can say to have a vaccine, at least for many of the approaches, you need to have, for instance, a very clear disease-driving antigen. For instance, there are vaccines against HPV-induced cancers. You can say the hypothesis and also the evidence is pretty clear that would work. Then what we see is of course a whole wave of what we can call personalized vaccines based on mRNA technology that we know from the COVID vaccine, where you take out, for instance, biopsies from the tumor, you analyze that and say what genes have changed here and which of them do we believe to be disease-driving and immunogenic, and then you base an mRNA vaccine on that. That will also work in a number of situations. What we see with our lead asset, mitazalimab, is that is a wonderful agent on its own in combination with chemotherapy, but we also believe that it will be a very good combination partner with vaccines. What the molecule does is that it does not activate T cells as sort of the effector cells that kills the tumor. We don't sort of activate those directly, but we activate sort of the underlying part of the immune system that both educates, attracts, and activates these T cells. So we have an opportunity to really boost the immunity or kickstart the immune system of the tumor. And especially tumors like pancreatic cancer, which are very, very few T cells, but very many of these, what we call myeloid cells, is a prime target for a drug like mitazalimab, where you can say other tumor types like melanoma are what we call hot tumors, so they have an ongoing immune response on an ongoing basis. Here, and now I'm diverting a little bit from your question, but I'm just getting so excited with the potential of what we are doing here, that in these inflamed tumors, as we call them, or hot tumors, where you have ongoing immunity, there is a risk that you sort of exhaust the immune system. You have ongoing immune responses, and really the body it sounds counterintuitive, but the body actually tries to regulate that immune response so it's not sort of too harsh or too effective. And our drug can actually take away some of those brakes on the system in those conditions. So it can be a speeder, an accelerator when needed, and a little bit of a dampener of other things when that is needed.
I see. Yeah. With regards to research, have you got good partners across Europe? Who are you using as your partner?
Yeah, so we have many good partners. First of all, we are fortunate enough to be part of a very strong research community here at Lund University with the immune technology, and if you look through our legacy and recent publications, you will see that reflected. There are very strong local collaboration there, which is, by the way, also a very strong base for research talent. We have a host of people that has graduated there and now help us on our mission to develop these drugs. We work with a number of institutions around Europe. We work with Erasmus in Rotterdam on a trial there, where we actually combine with a cancer vaccine. Now that we talked about that before, I would say when we talk about mitazalimab, then one of our prime collaborations is with the University of Pennsylvania, who has a long-standing record in the mechanisms that we are exploring and pancreatic cancer, and that has shown to be very fruitful. I think, as you rightly say, this is a team sport. It's a team of people. It's a team of companies and institutions. Yeah.
It is a team sport. I couldn't agree more.
Yeah, I think one of the things, and now we talked about mitazalimab and what that can do in pancreatic cancer, and I'm sure it can also be explored and effective in other GI tumors like colorectal and gastric potentially. And as we also discussed, it will probably also have a role to play in melanoma and other indications when we have the time and resources to explore that.
Are you thinking of repurposing that particular drug?
Yeah, you can say maybe it's not a repurposing. It's more sort of really getting it into indications where all the biology that and evidence that we have gathered over the last 4 or 5 years could actually become a benefit in those indications. So it's more sort of an indication expansion, I would say, than a repurposing.
And in Europe, I know, or Europe and the world, I should say, you've successfully achieved multiple patents for a number of different compounds. Yeah. Is there anything that you're working on currently that you'd like to talk about?
Yeah, and I'm glad you asked that, because that's something where I think we're really where we're really at the forefront. Not that we're not at the forefront with mitazalimab, that's in phase 2. So of course we are at the forefront there. But my very clever colleagues have developed a new concept which we call Neo-X Prime, and that's basically everything we just discussed with mitazalimab. So there's the binding to the CD40 on dendritic cells and activating them, and so on and so forth. But what we've done here is that we then sort of made these molecules that are normally bivalent. We made them tetravalent, so they have now 4 binding capacities. So they bind to everything that mitazalimab, the lead compound, does. And then they bind to so-called tumor-associated antigens. So that's antigens that are preferentially or exclusively expressed on tumors. And what that allows us to do now is, first of all, we get a molecule now that is only active in the tumor. So when CD40 on dendritic cells is there and the tumor-associated antigen is present, only when you have those 2 components together, you will get activation. So that gives you a lot of safety. Then in addition to that, the signal that we send into the immune system is much stronger, so we get much more sort of bang for the buck. And then finally, and I think this is the stroke of genius, by binding to these tumor-associated antigen, we now ensure that we take parts of the tumor and more or less sort of a UPS or DHL deliver it directly into the dendritic cell. And remember what I said, that these cells educate, activate, and attract T cells to the tumor. So what's happening now is that the immune system is actually now having a Xerox copy of what's going on in the tumor cell, more or less in real time, and is now starting to educate the T cells in the tumor on those neoantigens. Back to the vaccine we talked about, where you just had to take it out of the patients and sort of analyze it and then make a vaccine. We're actually making that happen on site in real time inside the patient's tumor. And I can tell you some of the data that we see right now, and we published some of it recently at a conference in the US. I mean, it shows so beautifully that we can now, with a single agent, very, very efficacious, eliminate tumors only with this molecule, without any adjunct therapy, any chemotherapy, whatever. But what we also show is that we can start killing tumors that only have this tumor-associated antigen expressed on some of the cells. The tumors will normally, when you put a selective pressure on the tumor cell, of course, start to downregulate these tumor-associated antigens. You see that in, for instance, in hematological malignancies, where you have antibodies against CD19, then the tumors downregulate CD19, then you have to give them CD20, CD22, so on and so forth. What we've seen in these models is that that doesn't happen here, because we give the immune system an opportunity to react to the entire genetic makeup of the tumor cell, so the entirety of the mutations. And now you actually get T cells that kill in a tumor-specific manner, but irrespectively of whether they associate or express this targeting molecule. So that's very, very clever. And we really see this as the next big thing in antibody-based immune therapy.
That actually sounds a lot better than vaccines, really.
It has its advantages, and number one is that you still do it personalized, but you don't have to wait 3-4 weeks or whatever it takes, or 5 weeks or 6 weeks to make a vaccine. You can actually treat on day one at diagnosis, and we see very strong data on that. And the strategy at Alligator is, of course, to develop distinct molecules that have that sort of binds to the signature molecule of distinct diseases. So we get very personalized, very indication-specific drugs that are highly effective and safe.
Fantastic. Thank you for sharing that with us.
That drug development takes a lot of time. It costs a heck of a lot of money, especially when you're getting into pivotal trials. Alligator is a relatively small biotech company, and we really want mitazalimab, our phase 2 asset, to benefit as many patients across applicable indications or relevant indications as soon as possible. We could definitely take mitazalimab forward in pancreatic cancer ourselves, but here the strategy is really to find a global partner that can, of course, ensure that the molecule is brought forward in pancreatic cancer, but potentially also in various other indications to get as broad as indication coverage as number one. So the near and midterm target here is to finalize the ongoing phase 2 trial, read out the data in the beginning of Q1, discuss with the Food and Drug Administration on what is the best way forward, and then use that momentum to really continue the dialogue we have with various partners on who is the best one to take mitazalimab further. And then take those proceeds from that transaction and really reinvest them in the concept that I just talked about before, to ensure that we can generate the next generation of really efficacious and safe drugs to benefit patients with these hard-to-treat cancers. So that's sort of the game plan for the next period of Alligator's life. I can tell you, I'm sure that it can be done, not only because we have the science, but because we have the guys that actually invented these molecules, the team here that allows us to run our trials efficaciously. I mean, we have so many brilliant people at Alligator, and I'm just so thankful to that entire team, because otherwise we would not be sitting here today, Stephen.
You're a good man, Soren, and I thank you very much for your time today. I'm very pleased we had this conversation. It's been enlightening for me, and I hope it has for the audience as well. I look forward to talking to you again in maybe 6 months when there's progress. And thank you for your time, and I wish you all the very best for the future.
Thank you, and it's been a pleasure, Stephen.